Hello everyone, welcome back to Microbial Engineering and Algal Biotechnology Laboratory, Department of Biotechnology, JIS University, Kolkata. The topic of deliberation of today is the vaccine and the drug repurposing against the COVID-19, the marks beginning to the end. The entire project has been conceptualized by Dr. Deepankar Ghosh, Assistant Professor and Principal Investigator of Microbial Engineering and Algal Biotechnology Laboratory, Department of Biotechnology, JIS University, Kolkata. First of all, I would like to acknowledge to me Brumi, especially my PhD research scholar Ms. Sheshtha Debnath and all Masters Research Project students, especially Sneha, Orpon, Momita, Tonni, Onkita, Shubhoshri, Dia and Rumi. The first segment of this presentation will deals with the vaccine against the COVID-19. So what is vaccine? And what are the vaccination procedures? A vaccine is a medical preparation given to provide immunity for a disease. Vaccines used a variety of different substances ranging from dead microorganisms to genetically engineered antigens or hapten to defend the body against the preferentially harmful pathogens. The effective vaccine alters the immune system by promoting the development of antibodies that can quickly effectively attack the disease-causing microorganisms when it enters the body, preventing disease development. A vaccine may contain live attenuated or killed microorganisms or the part or product of them capable of stimulating a specific immune responses comprised of the protective antibody and T-cell immunity. A viral vaccine should also be able to stimulate high titers of the neutralizing antibodies. Then what is the definition of the vaccination? Vaccination is defined as the immunization against the infectious diseases through the administration of vaccines for the production of active, that is the protective immunity in human or other animals. How vaccine works. The overview of steps leading to immunity after administration of a vaccine has been depicted over here. It shows very clearly that vaccine injection, when this vaccine has been uptaken by the system, it will induce either innate immune system or it will activate the macrophages. After activation and the migration, the signal will goes into the lymph node and that will follow up the presentation and activated. That is called the adaptive immune response and that will give rise to the cytotoxic T cells, plasma cells and the memory B or the T cells. So this is in one word says the clonal expansion and immunological memory. Then what are the various approaches for the vaccine development? There are 4 different segments are there. Number 1 is the live attenuated lab vaccine. Examples are tuberculosis, BCG, oral polio vaccine, OPV, measles, rotavirus and the yellow fever vaccine. Second segment is the inactivated killed antigen, that is the wholesale pertussis WP, and another example is inactivated poliovirus IPV. The third segment is the subunit purified antigen. Examples are as cellular pertussis AP, Haemophilus influenzae type B HIB, and pneumococcal PCV7, 10 and PCV13 and hepatitis B Hep B. The fourth segment is the toxoid inactivated toxin vaccines. Examples are tetanus toxoid and the diphtheria toxoids. So the next segment deals with the salient features of a vaccine antigen, which determines the subsequent levels and the duration of the immunity. This particular slide deals with the interaction between the B cell receptor, T cell receptor, protein antigen, polysaccharide antigen, PMSC class II molecules and cell-cell interaction. There are 3 different kinds of vaccine classes are there. Number 1 is the multivalent antigen. Example, measles virus particle, monovalent antigen, tetanus toxoid, multivalent non-protein antigen, that is Pneumovax 23. And these are the probable outcome. Number 1, increased BCR clustering, increased antigen presentation, increased dwell time with the TFH cells and long-term immunity. The second outcome would be the little or no BCR clustering, intermediate antigen presentation, shorter dwell time with the TFH cells and intermediate to long-term respectively. Now in the last segment, that is the multivalent non-protein antigen, which causes certain outcome, which includes the increased BCR clustering, no antigen presentation, no TFH cell help and the short-term immunity. Then a very important next question has come up, then how do different types of vaccine work against COVID-19? The first and most prominent one is the viral vaccine. At least 7 teams are developing vaccines using the virus itself in a weakened or inactivated form. Many existing vaccines are made in this way, such as those against the measles and the polio, but they require extensive safety testing. Sinovac Biotech in Beijing has started to test an inactivated version of SARS-CoV-2 in humans. The next segment is the viral vector vaccines. Around 25 research groups say they are working on viral vector vaccine. A virus such as measles or adenovirus is genetically engineered so that it can produce coronaviral proteins in the body. So these viruses are weakened so they cannot cause disease or pathogenesis. There are 2 types, those can still replicate within the cells and those that cannot because the key genes have already been disabled. Then the nucleic acid vaccine plays another important role. At least 20 teams are aiming to use the genetic instructions in the form of DNA or RNA for a coronaviral protein that prompts an immune response. The nucleic acid is inserted into the human cells, which then churn out copies of the viral protein. Most of these vaccines encode the virus's spike proteins. Then what about the protein-based vaccine? Many researchers want to inject coronavirus proteins directly into the body. Fragments of the proteins or the protein cells that mimic the coronavirus's outer coat can also be used. So this nice pictorial presentation shows that the efforts, which include the testing whether the existing vaccines against the poliovirus or tuberculosis could help to fight SARS-CoV-2 by eliminating or elucidating a general immune response rather than specific adaptive immunity, or whether certain immune cells could be genetically modified to target the SARS-CoV-2 virus. The next important segment that will be discussed, that is the repurposing and development of therapeutics against COVID-19. So this nice pictorial presentation deals with 2 different segments. Number 1 segment is the normal viral infection cycle, SARS-CoV-2. Second one dealing with the vaccination strategies. Viral life cycle of SARS-CoV-2 has been depicted over here by following few segments. Number 1 is the binding, followed by proteolysis, followed by replication, transcription complex development, translation assembly, and finally the virion particle get released by exocytosis procedure. Whereas on the right hand side pictorial segment shows that the vaccination strategies which are based on certain components which are present in the SARS-CoV-2, likely protein subunit, virus-like particles, VLPS, inactivated vaccine, mRNA vaccine, DNA vaccine, replicating and non-replicating viral vectors, followed by live attenuated vaccines. This current slide particularly deals with the different stage of their life cycle of SARS-CoV-2, and also it shows that what are the different proteins or the targets are involved where the different drugs can be repurposed. In a similar fashion, the next slide also dealing with the same manner, and it also dealing with in more clear way what are the different drugs, those who are going to be repurposed against the different stages and the target protein of the SARS-CoV-2 life cycles. So this is a typical example of the molecular farming which can be carried out in the tobacco and the maize plant. So this particular molecular farming approach, along with the synthetic pathway design and systems biology and heterologous operon expression, it shows that the SARS-CoV-2 neutralizing antibodies or the SARS-CoV-2 neutralizing lectin can also be developed, and that can be gone through for the large scale production in those particular plants. As I mentioned earlier, that these are the different categories of the vaccine that can be developed against the deadly virus COVID-19. That is the viral vectors, inactivated virus, subunit viral proteins, virus-like particles, DNA vaccine, followed by the attenuated viruses. Then how the developing a successful vaccine against the SARS-CoV-2? The unprecedented efforts are being made to develop a safe and effective vaccine against the SARS-CoV-2. Currently, scientists around the world are exploring all the potential strategies to develop an efficient vaccine against the SARS-CoV-2. This includes number 1, the live attenuated, followed by the viral vector-based, that is the S protein expressing, recombinant protein-based, that is the S protein spike protein, DNA-based, and the mRNA-based vaccines. Previously, it has been shown that the SARS-CoV-2 spike S protein is an ideal candidate for the vaccine development, as this should block the attachment and thus clear the virus in the early stages of the infection. An ideal vaccine must produce specific and efficient neutralizing antibodies against the SARS-CoV-2. However, the vaccines that can cause a number of unwanted immune-enhancing responses, such as antibody-dependent enhancement ADE of the viral uptake, the Th2 immunopathogenicity due to the faulty T cell responses which triggering a damaging allergic inflammatory responses. Usually, a classical vaccine development process takes longer time. Now, if you see this pictorial presentation, it clearly shows that the preclinical stage, which requires 18 to 30 months, phase 1, dozens of volunteers in 30 months, phase 3, the hundreds of volunteers requires in 32 months, followed by phase 3, thousands of volunteers which requires in 30 months. Finally, it will get approved, manufacture and vaccination come to the market, which overall requires 12 to 24 months. But in case of COVID-19, we require the clinical trial much more faster due to its huge severity and damage in the community. So it shows clearly preclinical trial stage doesn't require any time, doesn't have any time for us. Phase 1, the dozens of volunteers requires only in 6 months. Phase 2, hundreds of volunteers will be required in 6 months. Phase 3, thousands of volunteers, there will be no time left over. Finally, after just 6 months over in the phase 2, it should be require approval, manufacture, vaccination of billions of the doses and individuals only within a shorter period of time span of 6 months. The next segment will be dealing with some COVID-19 vaccine developing institutions or the research industries in the world. It shows there are 21 in the clinical evaluation and 139 in the preclinical evaluation. Viral vaccine or the virus-based vaccine has been developed by the Codagenics in the Flemingdale, New York in association with the Serum Institute of India, whereas Sinovac in the phase 3 trial, which is being developed, the virus vaccine by the Wuhan Institute of Biological Sciences, China. University Oxford, they are trying to develop the viral vector vaccines which are under the phase 3 trial. Cadila Healthcare Limited and the Moderna et al., they are trying to develop the nucleic acid based vaccine, whereas GSK, Novavax, in association with the Institute of Microbiology, they are trying to develop the protein based vaccine. Finally, the Medicago institution, they are trying to develop the protein based vaccine. So all this information has been taken from the Nature paper which has been published by the Callaway et al. Then what are the possible applications of the BCG vaccine to fight against the COVID-19? The Bacillus calmette-guerin vaccine, BCG vaccine designed to prevent the tuberculosis in children, has been shown to induce an adaptive immune response in the body to fight against the bacteria as well as other parasites and viruses. This knowledge has been reciprocated to generate the idea that this vaccine can also offer protection against the several acute respiratory syndrome, coronavirus 2 or SARS-CoV-2. Though it has not yet been approved by the WHO to use this vaccine to fight against the COVID-19 due to the absence of any firm evidence regarding its action, but it was adjudicated for the epidemiological studies that the BCG vaccination rendering the protection against the COVID-19 was because of the vaccine's non-specific effects. Reduction in the respiration tract infection in the children, antiviral effects, and reduce the viremia in experimental animals were attributed to the non-specific effects on the BCG vaccine. According to some possibilities hypothesized by the researchers that the BCG vaccine was able to prevent several respiration tract infections and reduce the mortality and morbidity by percentage as high as 70%. As BCG vaccine can hypothetically reduce the morbidity and mortality rate, it has also been administered in the body if healthcare worker with the direct patient contact during this epidemic phase in order to study the reduction and minimization in the action of COVID-19, to minimize the abscessism and to reduce the ICU and hospital admission or health in the workers. The impact of BCG vaccine in reducing the chances of respiration infection in the old individual. It has also been demonstrated in various controlled trials in the BCG vaccination reduces the severity of the infection by several viruses having a somewhat similar structure to that of SARS-CoV-2. The epidemiological studies has also been reported that the less occurrence of COVID-19 in BCG vaccinated infants are regulated by various influential factors. Those are the demographical differences, the burdens of diseases in the country, pandemic phase in each country, testing rates of the SARS-CoV-2 infection and each and every country. Now the current slide deals with the name of different vaccines in the international level along with their developers, their properties, the development status and the function. Now if you consider mRNA 1273, which is being developed by the Moderna and the NIAID, property mostly based on the mRNA vaccine. So in the developmental phase, they are in the phase 2. This kind of vaccine encoding for a perfusion stabilized form of a spike protein S. The S protein complex is necessary for the membrane fusion and the host cell infection and has been target for the vaccine against the coronavirus. The second vaccine is the BNT162 developed by the BioNTech and the Pfizer. Property mostly mRNA vaccine. Developmental status is phase 1 or in between phase 2. The RDB or the RBD based candidates contain the piece of spike that is thought to be most important for the elucidating antibodies and inactivate the virus. The next virus is the INO 4800. The manufacturer is the Inovio Pharmaceuticals, Nature DNA vaccine under phase 1 trial. INO 4800 targets the major surface antigen spike protein of SARS-CoV-2 viruses. The neutralization assay in the 3-phase show the vaccine candidate virus neutralizing the activity and ability to produce antibody. Then the next one is the ChAdOx, the INCOV-19. The developer is the Vaccines Group Clinical Team and University of Oxford and Jenner Institution. These are the type adenoviruses phase 1 trial it is currently in, and by vaccinating development and immune response to the spike protein that will help stop the SARS-CoV-2 virus from entering the human cells and prevent further infection. The next one is the NVX-CoV2373. The developer is the Novavax. Nature, it contains a small part of the coronavirus called the spike protein. It is under the phase 3 trial, large-scale clinical trial funded by CEPI. The major goal is to trick the immune system into thinking that the spike is actually the entire virus, thus creating a response for the attacking the real virus. The last one is the AD5nCoV. Developer is the CanSino Biologics and the Beijing Institute of Technology. Genetically engineered replication defective adenoviral type 5 vector to express the SARS-CoV-2 spike protein. It is under phase 1 trial. The vector delivers genetic material that encode for the spike SARS-CoV-2 protein to cell, which produces spike protein and travel to lymph node creating antibodies that will recognize the spike protein and fight off the coronaviruses. So that was like what people have done in the entire global arena. Now what attempt has been done on the COVID-19 towards vaccine development in India? The Bharat Biotech, a Hyderabad-based company, India has made the first possible indigenous vaccine against COVID-19, that is the Covaxin. The technical name given to this vaccine is BBV152 COVID vaccine. This vaccine was made with the collaboration with the Indian Council of Medical Research ICMR and National Institute of Virology NIV. The permission from the Drug Controller General of India to carry out phase 1 phase 2 trial in human clinical trial was based on the safety and efficacy results of studies on mice, rat and rabbit. Another potential candidate for vaccine production in India is the Ahmedabad-based company Zydus Cadila Healthcare Limited. ZyCoV-D has successfully completed a preclinical phase and has received permission from Indian authorities to conduct human trial. ZyCoV-D is a DNA vaccine which is very safe because it uses non-replicating and non-integrating plasmid carrying the gene of interest. Now the very important question comes, who would be the first to get a COVID-19 vaccine? As Russia ruled out its first coronavirus vaccine on the previous week, a panel of top Indian healthy experts is scheduled to meet to chart out the guideline to who should get the first injection and how to ensure adequate shocks of the vaccines. The Natural Expert Group of Vaccine Administration chaired by V.K. Paul, member of the NITI Aayog, is scheduled to deliberate upon the suitable vaccine for India and how to procure them and ensure the logistics for making it available throughout the country. In India, 2 vaccine candidates developed indigenously by Bharat Biotech in collaboration with Indian Council of Medical Research, Zydus Cadila Limited, has been phase 1 trial and has moved to phase 2, ICMR Director General Balram Bhargava said last week. The Oxford vaccine, expected to be manufactured by the Serum Institute of India, has received approval for the phase 2 and phase 3 trials. Russian President Vladimir Putin has launched a COVID-19 vaccine for the first time, touted as the world's first such vaccine, with one of his 2 daughters receiving the first dose. Russia has become the first country in the world to approve the vaccine for the coronavirus SARS-CoV-2. President Putin announced in the last week through the Global Health Authority say the vaccine has yet to complete late-stage clinical trial to determine its safety and the effectiveness as well. Britain and US has invested in a vaccine candidate being developed by the Oxford University and produced by the AstraZeneca. If it works, the UK politician has said the Britain will be vaccinated with it. About a dozen different vaccines are in various stages of the testing worldwide, including the Britain, China, US. This week, Dr. Anthony Fauci, the top US infectious disease expert, he said that he is cautiously optimistic there will be a COVID-19 vaccine by end of the year or early 2021. Positive news on the initial trials of the University of Oxford potential COVID-19 vaccine that has been licensed to the AstraZeneca. The potential vaccine is already in large-scale phase 3 human trial to assess whether it can protect against the COVID-19 or not. That is a big question. Now, what are the approval for the first coronavirus vaccine? This is a nice pictorial presentation which shows that more than 165 coronavirus vaccines are under the development. So vaccines, testing, safety and doses in the phase 1, 19 vaccines are there. Phase 2, 11 are there for the vaccines in expanded safety trial. Phase 3, there are 8 vaccines are in run, the vaccine in the large scale efficacy test. And finally, for the approval stages, there are 2 vaccines are there for the approved for the early or the limited uses. Now this particular segment is very important. Locally developed vaccine for the COVID-19 has been given regulation approval after less than 2 months of testing on human in Russia. Russian higher officials said the vaccine has passed all the required checks, adding that his daughter, particularly president's daughter, and has already been given it. The WHO said it had been a talk with the Russian authority about the undertaking a review of the vaccine, which has been named the Sputnik V. Currently, the Russian vaccine is not among the WHO's list of the 6 vaccines that have reached the phase 3 clinical trial, which involved more widespread testing in human beings. Despite rapid progress, most experts think any vaccine would not become widely available until mid-2021. Officials said that the Russian regulator has approved a COVID-19 vaccine developed by the Research Institute of Epidemiology and Microbiology in Moscow, even though the phase 3 trials of the vaccine had yet to be completed. This particular vaccine has been given to 76 volunteers as a part of the 2 early stage trials listed on the clinical trials government, but no results from those trials or other preclinical trial studies have been published. A little else is known about the experimental vaccination as well. The immunization is the first approved for the widespread use, but could be dangerous because it has not been tested in the large trial, says researchers. So this particular report has been taken from the Nature article published in 11th August 2020. Besides these different clinical trials, the next segment will be dealing with the drug repurposing to combat COVID-19. Then let's see what is the drug repurposing. Drug repurposing is the strategy for identifying new uses for the approved or the investigational drug that are outside the scope of the origin medical indication. Repurposed drugs are the immediate response to a crisis which has accelerated beyond the containment strategies. It is a strategy often used in the oncology space and the borrowing drugs from the other indications in a time severe because these drugs have proven themselves to be safe in the preclinical and the early clinical testing. In simple terms, it involves the identifying existing compounds through the biological plausibility in vitro, in vivo, in silico studies and clinical observations. Then what are the drug repurposing strategies for COVID-19 SARS-CoV-2? This is a nice pictorial presentation which shows how drug can be repurposed. Number 1 is the formulation, determination of the doses, combination or multitude of the drugs, delivery, therapeutics, orphan drugs, the failure candidate, the current candidate and approved drugs, and different pathway and their corresponding targets. So this is a nice pictorial presentation which very clearly shows that how the drug will be repurposed and what are their consecutive strategies for the SARS-CoV-2. There are currently no effective drug targeting SARS-CoV-2, the causative agents of the COVID-19. The first strategy for treatment of the COVID-19 is the repurposing of existing drugs that have already been shown to be safe in humans. The therapeutic agent against the SARS-CoV-2 exists at various stages of the infection, including one viral attachment through the ACE2 receptor, membrane fusion by the TMPRESS2 protease, the viral entry by the endocytosis, viral proteolysis by 3LP-Pro and PL-Pro, viral replications by the replication complex including the RDRP, followed by the host cytokine response interleukin 6R. Globally, there are many clinical trials underway to evaluate the efficiency of a number of repurposed drugs used individually or in combination against SARS-CoV-2, as well as the ability of the immunosuppressive drugs to ameliorate the clinical symptoms in severe COVID-19 patients. So this particular pictorial presentation is the proposed form, which is acting point of the anti-SARS-CoV-2 in replication cycle of the virus. In simpler way, I would say this is a particular kind of diagram where it shows different pathways and pathway targets which are being repurposed by different kind of conventional antiviral drugs. So let's discuss about the various existing drugs which are being used for the drug repurposing against COVID-19 SARS-CoV-2. So this tabular presentation shows that different drugs which are having different targets and their possible molecular mechanism against the COVID-19. However, these particular drugs having different approval for different infection curement. So if you consider the leponavir, ritonavir and darunavir, they have a possible mechanism is to inhibit the viral proteases. Their targets are mostly viral proteases 3LP-Pro and PL-Pro. And mostly they are approved for the uses against the HIV infection. Similar way, if you consider remdesivir or the favipiravir, these having target for the RDRP, and they are mostly used for the viral infection and Ebola viruses. And they have a possible mechanism for inaccurate viral RNA synthesis, and it may also block the viral nucleotide synthesis to stop or cease the viral replication. So similar way, there are so many different kinds of drug components are there which are approved for different diseases. Like if you consider chloroquine is mostly acts against the malaria. So it actually helps to treat the malaria. So these are anti-malarial drugs. Moreover, they have a target of the endosome and the ACE2 receptor. It can elevate the endosomal pH and interfere with the ACE2 glycosylation. Similar way, there are so many other drugs which are used for the rheumatoid arthritis, for the influenza infection or for protozoal infection curement, and they can also be repurposed because they have a similar kind of target like JAK kinase, S protein ACE2, and it has a molecular mechanism like it acts as an inhibitor that may disrupt the binding of the viral envelope protein to the host cells and prevent the viral entry to the target. Similar way, it may inhibit the viral protein expression as such. This is a typical pictorial presentation of the convalescence plasma therapy, which is the most important nowadays. So these are the strategy over here depicted as a nice pictorial presentation. Let's discuss about that. COVID-19 patient, when they gone for the recovery, then they are so-called the COVID-19 survivors. So you have to go for the screening of the COVID-19 survivors first out of the entirely infected patients. Now when you identify the COVID-19 survivor, you have to take the blood drawn, and from the blood you have to identify the virus neutralizing antibodies. This is mostly present in the serum. Then you can inject this particularly to the COVID-19 infected patients. That means for the treatment purposes, or you can also inject this to the healthy individuals as a preventive measure. Now let's discuss about the various companies and the industries, how they are investigating the COVID-19 drugs. So number one is the Glenmark Pharmaceutical. Investigating drug is the remdesivir. Their target, the Favipiravir inhibits the RNA-dependent RNA polymerase, that is RDRP, an enzyme that allows the coronavirus to replicate inside the host cells. Inside the cells, it is converted to the favipiravir ribofuranosyl 5-triphosphate and then blocks the RDRP enzyme of the SARS-CoV-2, the virus that causes COVID-19. The Glenmark Pharmaceutical has started the phase 3 clinical trial for the anti-flu drug favipiravir as a potential treatment for the patient suffering from COVID-19 in country India. Administration. The recommended doses duration has been fixed for 14 days. Day 1, a dose of 1800 milligrams twice a day is recommended by the Glenmark Pharmaceutical, followed by 800 milligrams twice a day for day 2 up to 14 days. The full 14 days treatment cycle will cost rupees 14,000 for one patient. The next phase is the Actemra investigating drug, that is tocilizumab. The target. This particular drug as a recombinant humanized anti-human interleukin-6 receptor monoclonal antibody can bind to the IL-6 receptor with high affinity, thus preventing the IL-6 itself from binding to its receptor, rendering it incapable of immune damage to the target cells and alleviating the inflammatory responses. Then what about the trials? The data from a clinical trial demonstrated the Roche unit Greenwich rheumatoid arthritis drug. This particular drug to treat serious ill COVID-19 patient was conducted by the Paris University Hospital Trust, involved 129 hospitalized patients with moderate or severe viral pneumonia. What about the administration? The first dose is 4 to 8 milligrams per kg. The recommended dose is 400 milligrams diluted to 100 ml with 0.9% normal saline, and the infusion time is more than 1 hour. For patients with poor initial efficacy, an additional application can be made after 12 hours. The dose is the same as before. The maximum number of times of administration is 2, and maximum dose of a single dose should not exceed 800 milligrams. The next most important investigating companies is the Gilead Sciences Inc. Investigating drug is remdesivir. Target. Researchers have demonstrated how the drug known as remdesivir works, preventing the viral RNA polymerase of coronaviruses as a target for these conditions. They found that the remdesivir blocks a particular enzyme that is required for the viral replication. Trial. The Gilead Sciences Inc. announced the top-line results from the phase 3 simple trial in hospitalized patients with the moderate COVID-19 pneumonia. The open-label study evaluated 5 days, 10 days courses of the investigational antiviral remdesivir plus standard of the care versus the standard of care alone. What about the administration? The patient must be injected with 200 milligrams of remdesivir on day 1 and 100 milligrams of the next 4 days. The Union Healthy Minister said an updated version of its clinical management protocol for the coronaviruses. Then let's think about the Merck. The Merck and Company, their investigating drug is the EIDD2801. The target, the active drug incorporation into the genome of the RNA viruses, leading to an accumulation of the mutation known as the viral error catastrophe or FORHAM. The trial, early stage clinical trial, phase 2, administration 200 milligram this particular drug, 300 milligrams EIDD2801. Another investigating institution is the Eli Lilly and Company. Investigating drug is the anti-COVID antibody. The target, it can mimic the immune response to the virus, for example by blocking so-called the spike protein that the coronavirus used to infect human cells. Trial on June 1st, Eli Lilly and Company moved to its expedition to the human trial. Administration base-wise, it's usually administered as infusions, but the process is complicated and capacity is very limited. They are also under investigation for proper uses and doses. After having a very deep discussion about the repurposing and all other stuffs, I would say this is a very novel technology people have been trying nowadays. This is not related with the repurposing, or the development of new vaccines, but little bit it is interesting, and I would say I would like to mention this particular approach and appreciate that one. Being published by the Rahman's Research Group in 2020. So this is nothing but the medicated nanofiber based mask development, which shows that it is the cotton plant fibers, those who are blended with the Indian medicinal plant extract. Then they go for the electrospinning process to develop the nanofibers. These particular fibers, they are incorporated into the mask. So this mask will be looking like the 3-layer mask, having the inner, middle and the outer layer. This particular 3-layer mask will be acts as a filtration unit, and which will minimize the mode of entry or first round of entry of the SARS-CoV-2 into our lungs and the upper respiratory tract. After having a long discussion on the drug developments, drug repurposing and vaccine development, let's discuss a little bit on medicines which are being recommended by Ministry of Ayush. Homeopathy, also known as the genus Epidemicus, this particular medicine was used during the H1N1 outbreak during 2008 and 2009, which was proved extremely beneficial. The pills consist of water memory of arsenic trioxide, otherwise a high toxic substance, which is being diluted to nullify its toxicity while using. Though Ministry of Ayush has recommended it for its uses during the COVID-19 outbreak, experts around the globe do not agree for its mass distribution. Ayurveda. In the wake of COVID-19 outbreak, the entire mankind across the globe is suffering enhancement of body's immunity and plays a very important role in maintaining the optimal health. Immunity promoting measures. Take Chyawanprash 10 grams in morning. Diabetes should also take sugar-free ones. Drink herbal tea or the katha made from the basil, cinnamon, black pepper, dry ginger, resin once or twice a day. Even you can apply golden milk, half a teaspoon of turmeric powder in 150 ml of hot milk once or twice a day. So I hope you will enjoy this, and if you try, you can make yourself and keep yourself healthy while you are staying at home. So we are almost coming to an end. Let's have a look on the current activities, those who are driven by MIB Laboratories under the supervision of Dr. Ghosh. Dr. Ghosh's MIB Laboratory is currently focusing on fetching out future novel drug targets against the COVID-19, especially on screening and identification of the novel therapeutics through the repurposing approaches and bioinformatic approaches, followed by metabolic engineering and synthetic biology against the novel targets selection for the COVID-19. So, scientists are still finding for effective and suitable vaccine candidates and also therapeutics for controlling the deadly COVID-19. So let's our hope never being end. As per quoted by World Health Organization, we need to follow this particular quote: be informed, be prepared, be smart, be safe, be ready to fight against SARS-CoV-2 COVID-19. Thank you very much for watching.